Hello, everyone, and welcome to today's live broadcast, Critical Appraisal of Animal Studies, presented by Dr. Annette O'Connor. We're excited to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. I am Sue Pham of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located at the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we can at the end of the presentation. Also, please notice that you are viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing this presentation, please click on the support button at the lower right, at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Now I'd like to introduce our speaker, Dr. Annette O'Connor. Dr. O'Connor received a Bachelor of Veterinary Science from the University of Sydney in 1993 and a Master of Veterinary Science from the University of Queensland in 1997, and a Doctoral of Veterinary Science from the University of Guelph in 2001. In 2009, Dr. O'Connor was admitted as a Fellow of Epidemiology to the Australian and New Zealand College of Veterinary Scientists. She is Professor of Epidemiology at Iowa State University, where she teaches epidemiology methods and inference in the Preventative Veterinary Medicine program. Dr. O'Connor currently works in the area of research synthesis, systematic reviews, and meta-analysis, and therefore devotes a large amount of time to critical appraisals of studies involving animals. Please join me in welcoming Dr. Annette O'Connor, and I'll now turn the presentation over to her.
Thanks very much for the introduction. It's great to be here, and I'm looking forward to giving the talk. So you've already had an introduction. I'm an epidemiologist by training, and at the moment I do a lot of critical appraisal of studies when we're doing systematic reviews, trying to decide what should we include in our body of work when we're doing that analysis. So what I wanted to do was start off with just giving you the agenda of what we're going to talk about. And I should say ahead of time, I have a little bit of a sore throat, so every so often I'm going to reach for my cup of tea. So we're going to talk about what's critical appraisal. Then we're going to make sure we differentiate between critical appraisal and comprehensive reporting, which are two very related but often confused aspects of critical appraisal. And then we're going to get into some specifics of actually walking through how to critically appraise a paper, focusing particularly on laboratory animal models. And the way we're going to do that is talk about the risk of bias domains. And I'm not sure how familiar people are with the risk of bias domains, but what it is a stylized way of thinking about critical appraisal. So that's the agenda that we're going to have, and we're going to start off with talking about a discussion of critical appraisal. So just to be familiar with some of the synonyms. So critical appraisal is a very old term. It's been around long since I started teaching critical appraisal. And one of the things about critical appraisal, I think, is that it's always been a bit of a mystery. You know, professors decide not to include a paper in a review, or they dismiss a paper, and they really articulate exactly the rationale for why the study is considered to be not of good evidentiary value and should be dismissed. And so critical appraisal can seem almost like a black box. And one of the other difficulties or problems with critical appraisal, it is often a judgment. And many scientists, especially early in their career, are really uncomfortable with the idea of making a judgment. They would like to have a fact-based approach. And unfortunately, critical appraisal is often a judgment, and there's no getting away from it. But what we would like is for the process or the rationale for our appraisal to become more transparent, because often I think historically it has not been. So here what I've got is a couple of synonyms for critical appraisal that we should be aware of. The term quality assessment is one that's been used more frequently in the systematic review arena, And it's an older term. If you think that systematic reviews have been around in the clinical sciences for maybe 20, 25 years, people used to do quality assessment. That's a very judgment-laden term that became quite unpopular. So you will see that, but it's becoming less used because of the value judgment that people found it offensive that a paper was considered to be low quality. The same with validity assessment is a term you might use, and I tend to use the word risk of bias assessment, but I think they're all synonyms, just so you're aware that these things are very much the same. So what it is an assessment of the validity of the results of a study based on how the study was conducted. So you look at how the study was conducted and you try to assess how valid are the results. And what's also important is you should make sure that if you're assessing a bias, which we'll discuss is do you think it's going to have a huge impact? Because all studies have some imperfection, right? So we're not looking for perfect. What we're looking for is egregious biases that we think are really going to change the inference. And then what we have to think about is what the direction of inference is. So, for example, are we more likely to reject the null hypothesis because of the way the study was conducted, or are we less likely? Because those things make a difference as we're assessing them. So, why do we have to do critical appraisal in the first place? Well, we have to do critical appraisal because not all studies are of equal value with respect to the decision we're trying to make. So some studies, when we read a study, we're trying to decide what will we do with this information. And maybe we're deciding to approve a drug. Maybe we're deciding to do the next process, the next study, test the next hypothesis in the scientific process. And not all studies are created equal. And we want to make the best decision we can. And therefore, we would like to use unbiased. We'd like to use unbiased answers, but really what we'd probably like to use is least biased answers. It's an essential part of the scientific process. And as I've mentioned earlier, and I've made this point here, often I think previously, critical appraisal has been potentially very informal, and sometimes it's kind of seemed arbitrary. So we'd like to move away from that approach. And, by the end of the talk, what we'd like you to have is one tool, and there are others that are available, that you can use to systematically, critically appraise a study and then make your judgment. And then also have your rationale for your judgment. So next thing I want to talk about is the difference between critical appraisal and comprehensive reporting, because I know there's been some work on comprehensive reporting, and I think tomorrow you're going to hear from Malcolm, and perhaps even today you heard from others talking about how important comprehensive reporting is. So let's talk about what critical appraisal is not. So critical appraisal is not the same as assessing whether a study is comprehensively reported. You'd be aware, I'm sure, that there are several guidelines available to, and I've got a few here, the ARRIVE guidelines and also the guidelines from the National Research Council on how to report the conduct and the results of an animal experiment. And these guidelines are a critical, a really key part of critical appraisal, because we need comprehensive reporting. We need to know what people did, and we need to know what people found to be able to critically appraise. However, one thing I do see is sometimes people use, particularly the ARRIVE guidelines, which is a checklist of, these are the things that you should report. And people sometimes use those as a critical appraisal tool. So a study that might, they count up how many of the ARRIVE guideline items are reported. Now, that does not equate to critical appraisal. Of course, we'd like to have all the items on the checklist, but just because an item reports, a study reports all the items on the ARRIVE checklist, doesn't mean it has a low bias. the authors might have done dreadful things. They're just very honest about it, right? They might have used a terrible approach to the study, but they're very honest about it. So we should not equate comprehensive reporting with critical appraisal. Although we need comprehensive reporting. So one of the things about these the reporting checklists is usually what you're doing is you're assessing whether the presence or absence of something being reported. But that doesn't necessarily imply biases. So let's have a look here. Here's an example of a text somebody might write. So on this slide, what we have is a piece of text that somebody might have written, and it'll say animals eligible for the study were randomized to treatment group using a random number allocation. So I assume by today there's been several discussions about the importance of randomization for reducing selection bias. So if we were using something like the ARRIVE guidelines, we would say that yes, check the box, the authors did report randomization. However, let's just have a look. You can see in the blue box, what I have is some baseline data about that study. So you'll see that I have the treated group, and there are 6 animals in that group, and I have the untreated group, and there are 6 animals in that group. So let's just imagine this is a 2-arm parallel study. So it's an experimental study, and it's got 2 arms, the treated and the untreated. Now, what we have here is the baseline distribution of sex at the beginning of the study. And what you can see here is that they have these investigators ended up, even though using random allocation, with 5 males in one group and 2 in the other group. So if we thought that that males were a potential confounder, as in they were associated with the outcome, then we might say that actually based on this baseline data, we have the potential for a selection bias. But both of these are examples of good reporting. So the authors reported that they did randomize, and they used a random number generated to do it, so we know that there's a check for a checklist. And here the authors have reported baseline demographics of the 2 treatment groups. So if we were using something like the ARRIVE checklist, we would give this a check for reporting approach to allocation. We'd give it a check for reporting baseline demographics. But from a critical appraisal point of view, we're probably going to reach the conclusion that there's a high risk of bias if sex is associated with the outcome, because we won't be able to tell if the outcome is caused, if any difference we see between groups, we won't be able to tell if that's because they were treated or because the majority of them were male. So again, here's another example. I think this is a really critical point, so I'm going to make it a few times. Just because someone reports something and they get a check in a reporting guideline does not mean that it's a low risk of bias. So for example, here we have that the authors have reported, as they should, how they identified the animals. And so you can see here down, the animals were with the treatment were identified with markers, and it might be something along these lines. So they have the sheep, that they've got an identifier, or you see the mice have a different identifier. Now, first of all, that's going to be a check for the ARRIVE guidelines. Did the authors tell you about the approach to identifying the animals? Now, the next step, which is critical appraisal of whether that's important, right? Is there a high risk of bias? Is actually going to be dependent upon something completely different. So for example, let's just say the outcome, we've got our blue box there, and I'm presenting the data again, and instead of this time reporting the baseline data as I did when I reported previously, now I've got the outcome. So let's say the outcome is mortality. Now, mortality is an outcome that's fairly hard to misclassify. Most times it's quite obvious if an animal is dead, and it's quite obvious if an animal is alive. And so the fact that you know that a mouse has a red dot on its head or a blue stripe on its back is not likely to introduce a bias. Again, the difference between a checkbox for comprehensive reporting and critical appraisal. Now, the other thing I want to point out here that's different between critical appraisal and comprehensive reporting, and this is just a general issue, is that when we are looking at comprehensive reporting, there are a lot of comprehensive reporting guidelines, as I've pointed out too. And the truth is that reporting guidelines are much easier to produce and disseminate than critical appraisal guidelines. And the reason for that is reporting guidelines are usually about a particular type of question. For example, the investigators might be asking about an intervention, or, so they want to know whether this particular drug has an effect on an outcome. And it doesn't matter what the drug is, and it doesn't matter what the outcome is. The study design is almost certainly going to be the same. It's usually it's a two-arm parallel design, or it could be a factorial design, but it's still parallel, or it could be a crossover design. But generally the design is the same. And we know how to conduct that design to reduce bias, usually quite well. And that goes across all disciplines. So comprehensive reporting guidelines tend to be about a particular study. So, for example, about an experiment, a parallel experiment, or a randomized controlled trial. Or you might get comprehensive reporting guidelines for how to evaluate a diagnostic test. On the other hand, when you get to critically appraising, you'll find a lot fewer, because critical appraisal usually requires expertise that is topic specific, because you need to know you need to know what factors are associated with the outcome. Because if we look back here at our two groups, what we need to know is what factors could differ between the groups that could affect the outcome. And that often requires in-depth knowledge about the outcome and in-depth knowledge about the treatments. And therefore, usually if you're doing critical appraisal, you have to take the domains that we're talking about and think, how would they apply to this topic? So that's another difference between critical appraisal and comprehensive reporting. And for that reason, this comment that I've got here is that critical appraisals need topic-specific expertise, and therefore there are very few canned critical appraisal tools. You usually need to have to go and identify what are the potential confounders. How could somebody misclassify this? You need to know what are the confounders. You also need to know, for example, for the outcome, how are you going to measure the outcome in this particular area, and how can that go wrong? And so just to make the final point about this, so this is continuing this example that we had about the comprehensive reporting and how you need expertise for critical appraisal. So now what I've got the comprehensive reporting. So the authors have told us about how they identified the animals. But now I've got two outcomes. I've got mortality. And as we've mentioned, mortality, it's going to be difficult to misclassify that, right? But on the other hand, if I'm studying a different outcome, perhaps in this group of animals, I'm studying companionship behavior, which is a very subjective outcome. So for that situation, I probably need an expert to tell me, how can I accurately measure that, and how can that be poorly measured, so that when I read the papers, I can assess whether the authors have an accurate measure of companionship behavior, or do they have a poor measure of companionship behavior, because that will affect my critical appraisal. So now I want to talk about the actual components of critical appraisal and how we're going to do it. And we're going to start off by talking very quickly about external validity, and then moving rapidly on to internal validity. because internal validity is what matters. But we should mention the concept of external validity, especially with respect to experimental animals. External validity is often sometimes called generalizability, although there are other terms, there are other uses for the word generalizability, so it can be problematic to use that term. So external validity is about who you can extrapolate the results of the study to. And the problem, it's really hard to critically appraise that, because who you can extrapolate the results to is in the eye of the beholder. And we know that in laboratory animal experiments, sometimes we feel that the results of the findings can be extrapolated to humans, and that's why we do laboratory animal models, because we think that they're a good model of behavior or outcomes in humans. Or perhaps we're more interested in extrapolating to the next population, the next research population. So external validity is a judgment call, and we're not going to spend much more time on it. What we're interested in mainly in critical appraisal is internal validity, which is, do the results of this study represent what actually happened in the population, the source population? And in laboratory animals, the idea of a source population is difficult to understand. So I think a more pragmatic approach to thinking about internal validity is, are the results of the finding valid, or were they influenced by the way the study was conducted? So let's just talk more about internal validity. So this is the one that we're really interested in. Now, I should point out that we're really interested in internal validity because if the results of the study do not represent what happens in mice generally. So for example, if you do a study on Dawson mice, 20 of them, and those results cannot be even extrapolated to other Dawson mice or your colony or whoever they came from, then they're really pointless, right? There's not much you can do with them after that. So external validity is a moot point. So we always spend our time initially thinking about internal validity, and then we make some assessment about external validity. So just a little bit of a refresher. Hopefully this is not too basic, but first of all, what do we mean by the results? So I'm going to think quantitatively. So let's just start here. We've got two groups of mice, and we've got the group of mice who are bounded by the blue box, right? So it's these guys. So they're receiving one treatment. And then we've got another group of mice that are receiving whatever's in the red treatment, right? So what we want to do is measure our outcome. And here you can see that I have a categorical outcome. And what it does is something makes the mice go dark and get pink ears. So you can see the outcome occurred in 2 of the 10 mice here and 4 of the 10 mice over here. So this is really basic stuff, although I managed to get it wrong. So how am I going to summarize the results with one number? And there are 2 ways I'm going to do that. One is I could do the risk difference. So I'm just going to subtract them. So the risk of getting pink ears in the red group is 40. The risk of getting pink ears in the blue group is 20. And you can see I've got a typo here. The difference should actually be 20. So that's one way I can get one number to describe the results of my study. Now, the other way I could do it is I could just calculate the ratio of those. And as an aside, generally when we have categorical outcomes like this, so this is you're in a category, we usually prefer to summarize the results using a ratio, right? So here I've got 20 divided by 10. You've got a risk ratio of 2, which says you're twice as likely to get pink ears if you're in the red group than the blue group. So why do we, just as an aside, why do we do it with the ratio? Now, because we don't want the outcome to be dependent upon the prevalence. So here, what I've got here is the same experiment, except for now I've got a larger sample size. And I have the same number of outcomes. So you can see here I've got 2 out of 15 and 4 out of 15. Now, if I calculate the risk difference, now the risk difference is 13, whereas before it was 20. So same intervention, different number. But if I use the risk ratio, then I get 26 divided by 13, and I'm still able to say the red treatment doubled the risk of getting pink ears. So we like to have what are numbers that are called generalizable, and that's one of the reasons why I prefer not to use the word generalizable for external validity. This risk ratio is a number that should work across populations. Well, what we're really interested in critical appraisal is, did the observed ratio, that risk ratio there of 2, or the risk difference, that of 13, did it arise because of the treatment? Did I get that 2 because of the treatment? Did it arise because of systematic bias, something I did incorrectly in the study? Or did it arise because of imprecision? And that's basically critical appraisal in a nutshell. So now if you want to think about that a little bit more pragmatically, the other way to think about it is, what is the potential for any factors other than the treatment to have influenced that 2? To give us a 2, was there something else other than the treatment? Because what we'd like it to be is the treatment, because that's what we want to make inference about. And that is the idea of critical appraisal. No, so what are the ways that other factors could make it not the treatment? So there are 2 ways that this can happen. One is imprecision. We know that every experiment's result is just a random event, right? So if we have a very small experiment, we have a much higher potential for random error. And so one reason that we might have got a 2 when the real number was 1 could be just the way we sampled. We just took a small sample. Now, we can solve imprecision by sample size. Now, I just want to explain why small sample sizes leads to the potential to get a larger, a more imprecise estimate. And the example I'm going to use is not actually an intervention. I'm going to imagine that we are interested in estimating the number of women at the institution at which you work. And I said we've got 26 people, so we might have 26 institutions, and we would like to estimate what is the proportion of women. So that's our result. So if I sent you all out to sample at your institution, and you could take a random sample, so you've got a sampling frame that has all the people at the institution, but I only let you select 3 people. Now, when you do that, you have the potential to get 4 different numbers. You can get an estimate of 0%, 33%, 66%, and 100%. And I think most of us would think that, let's just imagine that the prevalence is actually 50%. Most of us would think that if we only took 3, that there is actually a chance that we would get 0 women in our sample, and there is actually a chance that we would get 3 women in our sample. So those extreme values, there we go, those extreme values are possible, right? It's very imprecise because it's extremely unlikely that any of us work at an institution where there are 0 women or 100% women. Now, on the other hand, if I tell you, look, you don't have to take 3, you can take 30 samples. Now the chances that you will get 0 women out of 30 in a population that has 50% women is very, very slim. And the same for 100. So your potential for imprecision decreases as your sample size goes up because your variation decreases. And of course, if I said to you, you could take 300 randomly selected faculty or whatever from your list, then the chances that you'll get 0 is almost infinitesimally small. So you can solve precision by increasing the sample size. But you should always remember that one reason that you could get this 2 is just, it's a random event, and you've just sampled a randomly rare group of animals, which is why we want replication, right? Because unless we have replication, we really don't know how random that event is. We rely on replication to help us understand, to reduce the potential for a random event. So the two things that reducing precision are increasing your sample size, but of course that's the basis for meta-analysis, is replicating. If we see people do a study over and over again and we get the same finding, then we start to think that must be the result. So the other reason that we can get the wrong number is because of systematic errors. Oh, she says. Systematic errors are errors that it doesn't matter what you do with the sample size, you're not going to correct the problem. The classic example of a systematic error is, well, the easiest to understand is, imagine that I'm interested in measuring the length of rats' tails for whatever reason. And so imagine this is my ruler, and I think that it is 10 centimeters long. But it turns out that it's actually 9.5 centimeters long. Now, that means that it doesn't matter whether I measure the tails of 10 mice or 100 mice. I am systematically going to underestimate the length of my population. And so that's a systematic error, and it doesn't matter what I do about it. It's going to happen, and it's irrespective of sample size. Okay, so we're going to focus on internal bias, internal validity. And now we're going to walk our way through what are the ways that we can think about the factors that could make differences in the groups occur other than the treatment. And I think this is what I really like about this, this approach to risk of bias, is that it gives you a set of factors to consider very explicitly. And in truth, many, many people are already using these. They just don't have a name attached to them. And I think this, this approach to risk of bias assessment is a real step forward. I would point out that the nomenclature of bias varies, but the one that I'm using is based on the Cochrane group, who do systematic reviews in human health of intervention studies, particularly randomized controlled trials in humans. And the terminology that I use is theirs. And the reason that we use theirs rather than others, because epidemiologists tend to use slightly different terms for risk of bias, is because in animal experimentation, the designs that you use to assess interventions or exposures are very similar to the designs used to assess interventions and exposures in people. So it's, the parallel intervention, the experiment, or the crossover design, or a factorial design if you're interested in more than, if you're interested in interaction. So because of the similarity between the assessment of interventions in people and the assessment of interventions and exposures in animals, it makes sense to use the nomenclature that the Cochrane group uses. And this is them. So now what we're going to do is work through these. And we're going to start off with probably one of the easiest to understand, and that is selection bias. So what we're looking at now is trying to think of, going back to that first question that said, what are the factors that could have explained my two other than the treatment? And this is them. One factor could be selection bias. One factor could be performance bias, detection bias, attrition bias, reporting bias. I've already talked a little bit about random error, and at the end I'm going to talk just quickly about multiplicity. So let's talk about selection bias as a factor that could have caused me to see a 2 when it wasn't actually a 2. So the way that we're going to do this is we're going to start off, we're going to have an example, and I'm just going to illustrate. Some of these are really egregious, to sort of really drive home the point of how this could provide an alternative explanation for the result, the risk ratio of 2. So just let me have a cup of tea. Right? Okay, so Selection bias. So here I've got a quote there in blue, and what I've said that mice in the study were allocated to the treatment group by sex. So treatment A consisted of female mice and treatment B consisted of male mice. So obviously the way that the investigators selected the animals for the treatment is based on sex. Now, we might think that that has the potential to cause the groups to be different with respect to our outcome. And that's actually the critical factor here. What you need to know is the factor that you're worried about is whether there's another factor at the baseline. So when you allocated the animals to groups, or when you're reading a paper and they're talking about how they allocated animals to the group, is there another factor at the baseline, and by baseline I mean at the beginning of the study, that could be related to the outcome that might explain the result? So here we go, we've got this experiment again, but this time what I'm going to do is you'll look at the next slide, and believe it or not, these five mice here, they're going to get fat, and these two mice here, they're going to get fat too. So our outcome is going to be weight gain over this period of this experiment. And so believe it or not, these mice get fatter, and what you can see is, whoop, look, there they go, they got fatter. So we've finished our experiment, and so we've got a few mice that gained weight in these group, and you can see a few mice that gained weight in this group. And of course, it wouldn't be surprising that our outcome would be weight gain, and so we would sum all these up and get the average weight gain here, sum all these up and get the average weight gain here, and then we would, because this is a continuous outcome, we're probably going to subtract them, not take the ratio. And so we're going to say that the way we're going to summarize the result is that the mean difference in the groups is 20 grams. So here's the mean in this group, here's the mean in this group, and the difference is 20 grams. And what we'd like to do is attribute that 20 grams to the treatment. So we'd say getting the blue treatment made you gain an extra 20 grams. Now, if I were to tell you at the baseline that instead I don't have sex evenly distributed across the groups. So if you look here, each of the animals that's in one of these little boxes, if I were to tell you that they were male, then you might say, hang on a second, there's a lot more males in this group than there are in this group. And we would expect males to have higher levels of gain. Then you would say that another reason that I might have seen this 20 grams of difference is not the treatment, but maybe it's the sex distribution. And so it's another factor. So the factor that you're interested in is sex. You think that sex is associated with the outcome. And the issue is, how did you select the animals to get into the study? So whatever approach was used here didn't balance this confounder, this third factor that's associated with the outcome evenly. So you're looking for a selection bias. What you're looking for, the bias occurs because of the way you selected the animals into the study. Now, are the groups comparable such that the observed difference is attributable to the treatment rather than the confounder? And the truth is, in this example, you can't tell. And the answer is likely no. It's quite possible that the difference that we saw is because of sex, not the treatment. Now, this is not a study design lecture, but of course it's worth talking about how one avoids selection bias, and what you're looking for in a paper to try to say, well, is this study likely to have a selection bias or not? And the most important factor is randomization. Now, one of the things that's problematic with randomization is that when you have small sample sizes, it is quite difficult to ensure that known confounders and unknown confounders are evenly balanced. So you would be looking to see if studies randomized. But if you had an important confounder like this, like sex, it's quite possible, even if you randomize with such a small study, that you could end up with this sort of uneven distribution. So what you might also look for is what's called block randomization or stratification. In which case, what you'd look for is for people to say, well, I'm going to take all 10 males, and within those 10 males, I'm going to randomize to treatment. And then I'm going to take all 10 females, and I'm going to randomize to treatment. And that way, I'll make sure that the males and the females are evenly distributed. So what you'll be looking for is things like randomization. You'll also be looking for the sample size, because if you've got 1,000 mice, then you don't need to block randomize. It's likely that you'll evenly distribute the confounders. If you've only got a very small study, then you'll be wanting to know, did they do anything extra in the paper to control for confounders, like blocking or stratifying? The other thing you'll be looking for if you're critically appraising is for them to report baseline demographics. Remember back at the beginning, I showed you a study that said, hey, at the beginning I had 5 mice that were male in one group and 1 in another. That reporting of the demographics of the mice with respect to important confounders is something that you're hoping that they have reported, and they've reported comprehensively. So when you're critically appraising, what you're looking for is that the two groups are comparable, and where you'll find that information is by looking for the allocation approach. Did they randomize? Did they block randomize? You're looking at the sample size to decide, do they need to do something extra control for the confounders? And you're looking for them to report the demographic information. Now, one of the things I would say that's often not considered very well in experimental studies is that restriction is one of the most common ways that laboratory studies control for confounders. So if I were to go back here and I said I only studied male mice, then clearly I'm not going to get confounded by sex. Now, of course, we can't do that anymore with respect to gender, but with respect to sex. But restriction is a really common way and effective way that laboratory animal studies control for selection bias. They just exclude them. I'm going to have animals of the same age, of the same genetic line. So that's the other thing that you should look at, is how much variation do you think can happen in this study population that you've got. So the next one I want to talk about is performance bias. So performance bias doesn't refer to at the beginning, because performance bias, so we talked about selection bias, which is at the beginning. You know, are the animals even with respect to at the beginning of the study? Well, now we're talking about performance bias, and that is sort of over the period of time of the conduct of study. Were there systematic differences in the care of the animals or the housing that could have created the difference? So think of performance bias as across the whole, for the length of the study. And, I have a really simple example here. Mice in the infected group were housed in the newer BL3 isolation facility in individual cages. However, as there was no biological need and to reduce costs, the untreated control mice were housed in 2 group cages in the older BL2 facility. Now, clearly there's a possibility that the difference between the 2 groups, especially in weight gain, if you go back to my example previously, might be attributable to A, the difference in facility, and B, the difference in housing. And so, as I said, again, what you're saying now is, now tell me about the whole conduct of the study, and is there anything in that time period that could have caused differences? So when you're reading a paper, what you want to know is, were they housed the same? Were they cared for in the same way? And I should point out, so there, in animal studies, we're really looking at 2 things: the housing, the bedding, those aspects, and the ARRIVE guidelines sort of say, tell us about that. The other thing we're looking for, which may or may be not relevant depending upon the topic, and hence it's topic specific, is whether the caregivers who are making the decisions about additional care or rations, etc., are aware of the animal's group. So that's an issue of blinding. So I do want to point out that blinding is a tool that we use to prevent bias. But we shouldn't use the words single, triple, and double in animal experiments. I would encourage you to look for papers or in your own writing to talk about what was blinded. So if you think about biases, one bias is performance bias. And what you'd like is for the caregivers to care for the two treatment groups evenly. So you should blind the caregivers to the status of the animal. So as I said, performance bias is about whether over the period of time, the way that you cared for the animals that they were housed could have caused the difference. And in my example, it could have caused a difference in weight gain. And what you're looking for is that sort of information, which should be in the methods and materials. The next sort of bias is detection bias. And this is the one that people are quite familiar with. So detection bias refers to the end of the study, as in now you're measuring the outcome. So we talked about selection bias, which is at the beginning of the baseline. Performance bias, which is across the whole edge of the whole study and the care. And now we're talking about detection bias. And I've got an example here. The cages of mice in the group treated with the fantastic new drug were labeled with the term fantastic drug. And the cages of the mice that did not receive the treatment were labeled with old drug. And each day, the staff feeding the mice were asked to describe how active the mice were on a scale of 1 to 5. So here what we're doing is we're assessing the outcome. So this is the end result. And clearly, because what we're doing is measuring the outcome, and there is likely to be a bias in the way we're measuring the outcome. Because in this example, the people who are measuring the outcome know the animal's treatment group. So here is another spot where we would blind. Now, previously we blinded to prevent performance bias. Now we're blinding to prevent detection bias because we want the outcome to be assessed without knowledge of the treatment. So again, this comes back to this issue of the difference between comprehensive reporting and bias assessment. So blinding is something that people can report, but that doesn't necessarily mean that the study is or isn't blind, right? So detection bias occurs when there are systematic differences between treatment groups in the way the outcome is assessed. Now, to decide if there's detection bias, you're going to have comprehensive reporting about how the outcome was measured and information about blinding. Another bias that we see that's a little bit harder to understand is attrition bias. And it's probably best understood by looking at this example. Attrition bias is loss to follow-up. So you lose animals in the study. Now in human health, this is hugely important because people can choose to leave a study. So there's voluntary attrition. I get fed up with this study. Or there's involuntary attrition. People die from competing causes. So of course there's no voluntary attrition bias in animal studies. It's all involuntary. But here's an example. So for example, here we've got 24 animals were allocated to 2 treatment groups. So we know we've got 12 per group. And what you can see here is the average weight of the mice in one group was 2.5 grams and N equals 7. And then it's 2.4 in the other, but N is equal to 12. So we can see that we've lost 5 animals. And so the question is, really, if we've lost 5 animals, is it really valid to be comparing these? And what was the result of those 5 animals? If we imagine, I'm going to try and draw this if I can, that we had a really bimodal, I don't think that's going to work. Yeah, no, that's not going to work. We won't bother with that. So if we imagine we had a really bimodal treatment. So the first group, there were one group of animals that were really healthy and another group that were really unwell. And they actually reached a weight loss point where we had to euthanize them. So in truth, if they'd have been allowed to survive, they would have had a very low weight. So, but we've removed them from the study for valid ethical reasons. So now what we're comparing only is the survivors in treatment group, the first treatment group. And those survivors might not have a weight that's representative of everybody who was in the study. And so that's an attrition bias. You got talking to a statistician about what's the outcome you should be measuring is probably the best way to think about attrition bias. In this example, survival would probably have been a more appropriate outcome than average weight gain, because in the first group, 5 animals didn't survive to the end. And last but not least, of the systematic biases, one that we should watch out for is reporting bias. And the way that we see this can be quite difficult because, again, we're hoping that people report comprehensively, and if they don't, we can't assess this. What you can see in this example is that the authors have reported that they've assessed the weights of liver, kidney, brain, spleen, and lungs. So that's what they measured. There, no, that didn't work. That's what they measured. But when the results were read, they've only given us the results for the kidneys. So there's a difference in the kidney weights, and that might be interesting, but there's no information about the brain, the spleen, and the lungs. And so this gives the impression that there might be a big difference. But if the brain, the spleen, and the lungs are actually not different, then maybe that is really important to us, because we'd said that 4 of the organs had no difference out of 5 might be more meaningful to us than just this difference. So this would be problematic from a reporting bias point of view, because it seems like the authors might be cherry picking the results and choosing to make all their inference on the kidneys and not the other information. Now, of course, there's nothing we can do about that, except for we might give the study less value. We might say, well, it seems like some results are hidden. And there's nothing you can do about it, but it might give you less confidence in the overall inference that the authors are making. I've already talked about the potential for random error and the fact that the bigger the sample size, the less likelihood that you have a random error. But I also do want to point out that when we look at the p-value, that gives us the probability of observing a difference as big as we saw or bigger under the null hypothesis that the results are the same. And when, so you can see here, we have an exact p-value, and you can see, wow, it's really unlikely that we would have seen this result, this 90 versus 10. So we say, well, it is quite a rare result. So it's either a big effect or we've got a really rare result. On the other hand, when authors report this way, just the p-value is less than 0.05, we really can't make any judgment about just how rare this result was. In this example, you could have done the data analysis yourself, but not normally. So maybe the p-value is 0.06 or 0.049. It's less than 0.05. Maybe we think, well, that's not that rare. Like if it's 0.049, that means it basically happens 1 in 20 times. And that's not that rare. Like get 2 die 1 in 36 times, and we don't think that's an incredibly rare event. So, you should hope that authors report comprehensively, and you can assess that. And then I want to talk about multiple testing, and you should think about, when you're critically appraising papers, multiple testing. Multiple testing, we know we're doing hypothesis tests, and we know that if we have, say, a p-value of 0.05, that means that we would reject the null hypothesis when it's true 1 in 20 times. So if you just do a ton of tests, then you're likely to end up with some statistically significant results just out of dumb luck. So here we have an example where we've got 3 treatment groups. We've got wet weight, dry weight, and surface area for a plethora of organs. And we're going to do all possible comparisons. And it turns out that this actually ends up being 63 different hypothesis tests. So really, you would expect some to be significant just by random chance. And so when you're looking at a paper and it's got just a ton of outcomes and only a few of them are significant, you might think, wow, these are potentially just random. So what to do about multiple testing is a whole 2-hour-long lecture in experimental studies. But you should seriously think about, if they're testing lots of outcomes and you've got a few things significant, maybe it's just random error. One of the problems with this is that people comes back to reporting bias. If the authors don't tell you that they tested 63 things, and they only tell you about 3 or 4, and then they report the results of those 3 or 4, then you can't assess the impact of multiplicity, which comes back to many of the issues of reproducible reporting and comprehensive reporting. So they're all the ways that you can assess bias. And I'm going to have to wrap up because I'm running out of time. But in the end, what I want to say is that you do have to reach a conclusion. And what we suggest is that you look through the paper that you're reading and you petition out the sources of bias. And you decide what you're going to consider to be low risk of bias, high risk of bias, and try not to use unclear. And I'm afraid to say that there is no way to avoid making a judgment. However, it's what we've always done, but now people are asking you to articulate why did you make that judgment. So you should say whether it's a low risk of bias or a high risk of bias, and it's not possible to avoid judgment. You're just going to have to do it. Do avoid giving scores. Do not add them up and say a paper has 5 highs and 2 lows. Avoid that because you might have, what you're giving is exactly equal value to each source of bias, and that's might not be appropriate. For example, maybe the detection bias is an absolutely egregious issue and makes the whole study invalid. And everything else is fine. But if you summed it up, that study would only get a point of 1. There we go. Whereas you might have another study that has, 2 other sources of bias. It has a multiplicity problem, but you're not that worried about it. So they might have a multiplicity problem in one study and a detection bias in another study, but you really think the detection bias issue is problematic. But if you sum them up, you're going to give them the same score, and that's not appropriate. So in our manuscript, we go through, how you would answer these questions and give it a high and low value. We also point out where you might look in a paper to find that information. And then we give you an example of how to do it. But I'm afraid to say that in the end, critical appraisal is always about judgment. However, here we enable you to petition up into these bias areas that I've discussed. I'll finish up now and point out that we have a manuscript that is freely available that goes through these topics, and at the end it has that checklist. And so I would encourage you to look at this if you're thinking about critically appraising a tool. Well, there's also another one by the Circle group, which is called Risk of Bias in Animal Experiments. It's very, very similar. But all of them now try to focus on petitioning how you critically appraise based on these different aspects of bias and trying to articulate how you reach decision of whether it was a high risk of bias and a low risk of bias, and nothing you do is going to get you away from having to make a judgment, I'm afraid. So thanks very much for your time, and I'm not sure I left time for questions, and if we do have any, we will have to answer them afterwards. I'll hand back over.
Well, thank you, Dr. O'Connor, for that very informative presentation. We certainly learned a lot from that. So it's now time for the Q&A. If you have a question that you'd like to ask Dr. O'Connor, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on the screen, and click on the send button. We'll answer as many questions as we have time for. So we have one question now for Annette, and the question is, how do you conclude the results if we face health issues in control groups of the experiment?
Well, thanks for that. Thanks for that question. You know, unfortunately, there's almost no solution to this problem. What I'm assuming is the issue is that you've got, let's imagine you've got a two-arm parallel design, and one of them is a control group, and then you have health issues in that group. For example, maybe some of them, well, we've got health issues, and so maybe they die, and that's one of your outcomes, or maybe they've lost weight. Now, if those health issues are unrelated to the outcome, there's really nothing you can do about that. And I think, actually, now that I think about it, I think this all depends on what your outcome is. So if your outcome is going to be affected by health issues, so for example, your outcome is weight gain, and you think that those health issues are unrelated to the treatment, then it's going to be confounded. On the other hand, if you think the health issues are actually part of the treatment effect, then you shouldn't have any problem. So really it depends on whether you think those health issues are part of the treatment effect or not. And and that would depend on your outcome.
Okay, we have time for another question. And Juliana asked, what statistical test is appropriate when you have only N of 3 animals per group and multiple groups?
Okay, so the question was, what statistical test is appropriate? So the answer to that is the statistical test that you would choose is not necessarily rated based on the number of animals. It's based on the type of outcome you have. So if you have a continuous outcome, then you're going to be trying to do something like a t-test. If you have a categorical outcome, such as whether the animal died or not, then you're going to be looking at doing something like a chi-square test. The issue that you have here of n equals to 3 is that you almost have no power to detect any difference. Doesn't matter whether it's a continuous outcome or a categorical outcome. So the statistical test of choice is based on the nature of the outcome. Whether you'd have any power to detect a difference when n is 3 depends on the variation, and it's going to be rare. And multiple groups. Truthfully, if you say, when I talked about the t-test, that's a comparison of two groups, a very simple comparison of two groups. If you have multiple groups, so for example, you have treatment A, B, and C, you're still, the test that you pick is going to be based on the nature of the outcome. So for example, if it's continuous, you're probably going to pick something like an ANOVA, or you might use a regression or something. Not a regression. Regression's too broad. But if you've got a categorical outcome, you're still probably going to use some sort of chi-square test. But again, the issue of the n equals 3 is not the factor that enables you to decide what statistical test you're going to do. That's dependent on the nature of the outcome that you're measuring. But n equals 3 means you're almost going to have no power to detect a difference. I hope that answers that question.
Okay, I think that's all the time we have now for questions. So I would like to once again thank Dr. O'Connor for her presentation. Do you have any final comments for us?
No, thanks very much. As I said, one of the sad things about critical appraisal is it still ends up being a judgment. But if we can make those judgments transparent and comprehensive and more systematic, I think we'll move the area of critical appraisal, will progress a great deal.
Well, thank you once again, Dr. O'Connor. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing until August of 2016. You will receive an email from LabRoots alerting you when this webcast is available for replay. Please feel free to share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye-bye.